So welcome everyone. It is a pleasure to see also some scientists and researchers here, and it's always a pleasure to be able to talk to the parents and to share some information on these syndromes with you. So when I was preparing this, it crossed my mind that it's actually my 10th anniversary of being an Argonaut. So I just briefly wanted to show you to say a few words about it. So I have known Finn since he was 7 days of age, because I was actually called at a neonatal intensive care unit because the human geneticist who was supposed to take care of Finn was ill on that day, and that's how I met him. And then we performed trio exome sequencing and identified this particular variant in NAGA2. At that time point, we were not sure that this is the cause. This is also Finn at the age of 11 months. And around the time that he was 3, Auntie and I were discussing how do we get and contact other families. And then I made this Facebook group. And I'm really happy today that out of this Facebook group, such a great Argonaut Alliance came out, and that you are connected, and that you basically don't need me anymore to connect you guys. Okay? So, and also what I wanted to say, I'm also very happy that my colleague Jessica Johansson and I recommended Antje to go to Köln, where they have this special program, which is called in German Auf die Beine, where they are using a special vibrating plate which strengthens the muscles. And this was actually the improvement which helped Finn to start walking. And then yesterday we made a nice photo of Finn and me. So it's like becoming a circle. But also from my point, I mean, starting from one patient, this has become one of the most important research projects in my group. So we deal with many different aspects of this disease. I also want to thank many of the researchers with whom we collaborate. So this being said, I will now say a few words about neurodevelopmental disorders. Or do you want, Amelie? I was able not to cry too much with the first slide. So, I mean, there has been a lot of discussion about the fact, about this various clinical symptoms that are observed in your children. And it does make a sense, because these neurodevelopmental disorders are actually a group of various symptoms which in various and different ways affect the function of the human brain. So there are some children who will have more attention deficits, or where hyperactivity will be up front. Some will have more, some will have less problems with the muscles, like muscle disorders. Some will have learning disabilities. Some will have problems in communicating with the other people. There is also autism spectrum disorder, like Amelie said in the morning. It's just one of the symptoms which comes in this diverse package of NDDs, of neurodevelopmental disorders. There is also intellectual disability or developmental disorder. So these are all symptoms, and each of the children is a kind of unique. And something that we should also never forget, that our genome consists of 20,000 genes and of many, many other parts of the DNA that we still do not understand. So nobody is made just of this one single variant. What is also important to say is that in the majority of the cases, the cause of this disease are de novo variants, which means they were neither inherited from one or the other parent. And I think it is very important to say that it is nothing that you guys did wrong, that you could have not done something wrong during the pregnancy or before the pregnancy. It is absolutely not your fault that your child has a de novo variant. because this is something that I get asked a lot. So what are these de novo mutations? These are actually genetic mutations which are present for the first time, and we believe that they occur when during the first cell division, so-called meiosis. At some instances, it can also be a so-called germline mosaicism, which means that either the man has this mutation is just a few of his sperms, or that the woman has the mutation just in a few of her eggs. But this is something that we cannot test. But this is also the reason that we in Germany offer prenatal testing if you are for your following pregnancies. Also what we did when Antje was pregnant. Okay, today we know that this spectrum of disorders is caused by thousands and thousands of genetic alterations. And you have to imagine we always carry 2 copies of the gene. And in the cases of Argonaute syndromes, it's only 1 gene copy that is affected. That's what we refer to in human genetics as a heterozygous variant. And such genes actually make at least 1 protein. So we have this AGO1 and AGO2 gene. They're on different chromosomes in the human DNA, but have a very similar function. And that is also the reason why they cause a very similar disorder. And the majority of these cases are actually autosomal dominant. The majority of the cases, the mutation occurs de novo. It's not inherited from either mother or the father. And this 1 mutant or altered gene copy is the cause of what we now know or call Argonaute syndromes. The human genome consists, as I previously said, of more than 20,000 genes, of 23 chromosome pairs. There are millions and millions of letters, so-called nucleotides. This is, it's always interesting to see that our whole body, the whole evolution consists actually of 4 letters. And it's incredible how from these 4 letters, such a complicated system like our human body is being built. And sometimes we want to know from you what is the exact variant that your child carries, because we want to know if the symptoms that your child has are similar to the other ones. We want to compare them. And that's why it's always important for us if you can tell us these amino acids. So this is something that each parent should learn, that there are several amino acids and how they are called. There are 20 of them. And also the position of the amino acid. Sure? Okay. So, okay, no. I just don't want to take everything away from you, okay? So, what are the most common symptoms? And as the word says it, the neurodevelopmental disorder, it is primarily the symptoms that affect the function of the human brain. We see intellectual disability in almost all of the cases, but there is a huge variability in the degree of affection. We see really mild cases, and we see severe cases. And luckily, through a lot of work, we are starting to realize what is the difference in the terms of the variant that was identified in the child between these mild and severe cases. Then what is also extremely common is muscle weakness, especially right after birth. And this muscle weakness mostly causes neonatal respiratory problems because the children are commonly too weak to breathe. They're also too weak to feed themselves, so they have to be mechanically ventilated. They have to be tube fed. This hypotonia also results mostly in the delay of acquiring motor abilities and free sitting and walking. But this is something that we can intervene with specialized physiotherapy, like this vibrating plates which strengthen the muscles. And we see in many cases seizures, and also Olena will be talking about this later today. Many of the children have a impaired speech development. Some hardly learn to speak. Some learn to speak at a much faster pace. And this is very individual. Then we have a whole range of different neurological abnormalities like autistic features, attention deficits, anxiety, aggressiveness. There's also sleeping disturbances. And I know how many of your children, that you are here, have also tried melatonin, because melatonin is something that is commonly given to children who have problems with sleeping. And we have, unfortunately, a few cases who are even resistant to melatonin. Sometimes we also see unspecific changes when we do so-called magnetic resonance imaging of the brain. And then there are some symptoms that there are some skeletal anomalies. In some cases, we also see heart anomalies. We also have several problems with the eyesight, myopia, hyperopia, and also strabism. And what we also identify, what is not here, but I presented this morning, that a certain degree of children are more prone to allergies and asthma. So I hope that by the end of the year we can start deep immune phenotyping to really analyze the different immune cells in your children. For this, we would need 1 milliliter of blood sent to a colleague in Hamburg who is specialized in doing such analysis. And so far, there have been numerous so-called missense variants identified in different parts of this gene. And you have to imagine that each of our genes has some specialized segments which are called domains, and most of these domains have somewhat different functions. And this is a part of the reason why we see such a variable clinical phenotype, because each of the variants affects the protein at the end in a slightly different manner. Similarly, we observe a broad variety of variants also in AGO2 gene. Again, this is likely the reason why not every children is the same. And also, we believe that also variants in AGO3 are causative. And one of the primary reasons why I believe in this is the fact that we actually see the same missense variants across all 3 genes. So we see the same amino acid altered in AGO1, AGO2, and AGO3 genes. But Amélie and I are working and trying to decipher this. Okay, so what are our major scientific questions? First, we want to understand the clinical course. This is the reason why we have been collecting clinical data, both from the clinicians over the world who contacted us, but also with the questionnaires that you have been sending to Olena, and also through Genida project from Amélie. And this is extremely important for us because we want to understand what are the symptoms that may occur later in the life as well. We also want to understand what is the differential impact of various missense variants identified on the clinical level. This is so-called genotype-phenotype correlation, meaning correlation between the missense variant identified and the clinical course of disease. Then also what especially Hans-Jürgen has been doing a lot is trying to understand which of the diverse functions of this protein are really affected by each of the variants. It's really a hard and tough work that he's been conducting. And also something that we are especially interested in, how do the variants affect brain development. So, regarding the clinical course, we have been doing not really interviews with family members, but asking them to fill in the questionnaires. If we had a chance, and if the parents provided this data to us, we were also happy to revise the clinical data. Some of you also provided images and also EEGs, electroencephalograms, especially to Amelie and her team. And there has also been a patient registry going on with Dr. Ielesicheva. And we have now included 60 children in this natural history study. We would like to finalize it by the end of the year. So please, those of you who have not sent us the filled-in questionnaires, please send it till the end of October. Because at this step now, we want to really do a deep dive into that data and to understand what are the next steps. In which direction do we need to go to understand this disease even better? Because this is the point where we actually need some extra specialists. Like we will probably need some special child psychologists, especially for the children who have moderate or mild diseases, to also be able to analyze the degree of the communication defects, anomalies that they have. As I previously said, we are in contact with a fantastic immunologist who is willing to do deep immune phenotyping. But we need to go to do a deep dive into the obtained data to see if we need any more specialists. And with this, I would like to invite Olena to give you an overview of her results regarding the natural history studies. Thank you.
This next.
Yeah, with this you go next, and with this you can show. Okay.
Hello, everyone. I'm happy to be here and present the result of our natural history study to the second time. Last year, I presented the first hour results in general overview. For this study, we have created a special questionnaire, and with this questionnaire, we are collecting data from our patients. And to this presentation, I have analyzed the data from 16 patients, 25 patients with AGO mutations from 24 families, and 35 patients with AGO2 mutations from 29 families. And our questionnaire includes detailed data from general information, age, country, and gender, and others, maternal-paternal history, obstetric and genetic background, delivery history, antepartum history, neonatal postnatal period, and with this data helped us to determine the main clinical spectrum, and the focus of my presentation today will be on main aspects, important aspects, this AGO1, AGO2 comparison, genotype-phenotype correlation, and epilepsy. Also, we have data from 18 countries. Age distribution, we have more than 80% children under 18 age. And parental health, we have no significant healthy issues from parents. And no relevant problems and significant from during previous pregnancy. And average maternal-paternal age is 30 and 32. And genetic background, majority mutations are de novo, but we have 5 cases with inherited mutation, 1 case in AGO group. AGO1 group, we have 1 family, son inherited the mutation from mild affected father, and we have 4 families with AGO2 mutations. 1 family, siblings, brother and sister inherited the mutations from mosaic mother with mild symptomatic. 1 family, siblings, sister and brother inherited this mutation from also mild symptomatic mother. And 1 family, brother and sister, siblings inherited from no symptomatic, unaffected mother. And we have 1 family that monozygotic twins inherited this mutation from no affected mother with mild symptomatic. And the first signs that we paid attention were during the pregnancy, but in the AGOA as fetal placental complication, and in AGOA1 group, we observed only 2 cases with fetal placental complication, 1 case with intrauterine growth restriction, and 1 case with abnormal amniotic fluid, and 3 cases with hypertension, and 4 cases with diabetes. But in AGO2 groups, we observed more frequent complication, 6 cases intrauterine growth restriction, 4 cases with abnormal amniotic fluid, and 1 cases decreased fetal movements, and 2 cases with increased nuclear translucency. And these finds could be indicate to the prenatal risk, but we need more and more cases and more information for this study. And this slide shows very important information about delivery history. As you can see, only 20% patients from AGO1 group had normal natural vaginal delivery, and only 5 patients, 15%, in AGO2 group had normal vaginal delivery. More than 50% cesarean section, but not all cesarean sections are pathological, were conducted due to pathological reasons. For example, cesarean section could conducted due to previous cesarean section or baby position and another cause. But I have counted this percent of pathological cesarean section. In AGO1 group, it were 6 cases, 24%, and in AGO2 group, 12 cases, 35%. And assisted and operative vaginal delivery are also pathological. And we have 35% in AGO2 group and 24% in AGO1 group, and in total, we can see that in AGOA2 group, 30% had normal delivery, and 70% of patients from AGOA2 group were born due to pathological delivery. And in AGOA1 group, half of patients have normal delivery, but it's a lot in any case. And neonatal complication in AGOA1 group, less cases and mostly mild. And in AGOA2 group, higher prevalence. And this table shows that about 90% of patients with AGOA2 mutation have neonatal complication, different neonatal complication. And about 80% have, in AGOA1 group, have neonatal complication. And we can see that, yes, abnormal muscle tone in both group almost same, but we can see that, for example, neonatal feeding difficulties in AGOA2 group about 17%, and in AGOA1 group 24%. 6 cases and 24 cases. And respiratory problems and another problems. And we can see that patients with AGOA2 mutation have more and frequency neonatal complication. And developmental skill. Both group shows developmental skill delays. And for example, these figures show this distribution, and this green line show the reference values. And we can see that both group show developmental skill delays, but in group of AGOA2, we observed that average age of achieving of developmental skills is higher. And this percentage shows that how many patients did not achieve these developmental skills. For example, 7 patients, 20%, did not achieve independent walking, and almost about 30% of patients did not achieve these skills, first words. And this percent in both group is same, and this percent patients with AGOA2 mutations, more patients did not achieve this developmental skill, independent walking. And you can see AGO protein domains and patients' variant distribution. In my presentation, among the patients that I have analyzed, we can see mostly mutations located in linker 1. And I have 4 cases in PATS, 1 case in linker 2, and 4 cases in PB domain. And with this slide, I would like to show the distribution, main clinical symptoms, and protein domains. Linker 1, we have 13 cases, and PATS domain and linker 2 together we have 6 cases, and PV domain we have 4 cases. And we can see that patients with linker 1 mutations mostly have motor developmental delay, gait abnormalities, skill developmental delay, speech delay. 6 patients are nonverbal, and 2 patients need wheelchair assistance, but one patient need only rollator. And these patients show severe phenotype, more severity than, for example, patients from PV domain. And this, I would like to show this comparison between patients with different domains. But we have patients in this group. We have 50 patients. For example, in this group, we have only 4 patients, and this data cannot be relevant, and we need more and more cases and more data. And agoprotein domains and patients variance distribution. In this, we have mostly mutations in L1 and L2, linker 1 and linker 2. And also we have 2 patients in N-terminal, only 1 patient in PADS, nobody in this mid, and 5-6 patients in PV domain. And this table with comparison, this table shows that each group show severity phenotype, and linker 1 and linker 2 show more severity phenotype. For example, in this group, from 10 patients, 7 patients have seizures. Almost all patients have motor delay, developmental delay, gait abnormalities, speech abnormalities, and other. And this slide show comparison between AGO1 and AGO2 patients, and we can see that average percentage is same in both groups, and this group have same clinical spectrum, but more patients with AGO2 mutations need wheelchair assistance, and we can see that patients with AGO2 mutations have more problems with systemic problems, with visual impairment, breathing problems, gastrointestinal disorders, and others. And this figure shows AGO1 protein domains and epilepsy status. This indicates that red mutation with epilepsy, dark red, this we have, for example, 7 patients with this mutation, but only 3 patients have or seizures activity. And these we have 2 patients with this mutation, and 1 of 2 patients have seizures, epilepsy. And this table show in detail this type of seizures. For example, patients with mutations, phenylalanine deletions in position 180. We have 7 patients, but only 3 patients have seizures. But 1 patient have seizures activity in encephalelectrography. 1 patient have only absence, and 1 patient have tonic focal and myoclonic. There are 3 different types of epilepsy, but all 3 patients have a different type of epilepsy, different phenotype, epilepsy phenotype. For example, we have 3 patients with these mutations, and 2 patients have epilepsy, and also they show different variable phenotype from convulsive episode to generalized tonic-clonic seizures. We can see that many of patients have only absence and, for example, febrile seizures. Distribution depends on domains and linkers. I want to show the distribution of type of seizures, age of begin, frequency, and this more detailed information. These patients with mutation in linker 1, and we can see this age of begin, different from 6 months to 16 years. And frequency and contribution factors, and 3 patients have seizures free after treatment. For example, 2 patients have resistance from medication. And this table shows also in other domains. For example, this case, age of begin, few months. And treatment, most common medication are clobazepam, levetiracetam, lamotrigine, and these commonly medications. And in one case, it was surgery treatment, but seizure is ongoing. About 13% of patients have seizure-free, and 15%, 2 patients have persistent epilepsy without improvement. And summary of epilepsy among patients with AGO1 mutations. Prevalence, 30% of patients have seizure activity or epilepsy. Age of onset highly variable from 6 months to 60 years, most about 3 years. And they have phenotype discordance. It means that same mutations with different epilepsy phenotype. Patients have same mutations, but they have different phenotype. And some patients with same mutations don't have any epilepsy and seizures. And seizures types very variable, from generalized to febrile and absence. And these AGO protein domains AGO2, and this epileptic status. We have two mutations in this domain, N domain, and both have epilepsy. And for example, we have 6 patients with these mutations in helix 7, in linker 2 located, and 3 patients. This is family mutations. I told we have 2 family with these mutations and 1 separate patient. And 3 patients with this mutation from 6 have epilepsy. And this variant, this distribution. And this interesting that phenotype, epilepsy phenotype in mutations, AGO2 mutations, mostly concordance. This means that patients with same mutations have a similar phenotype. For example, we have 6 patients with this mutation, but only 3 have epilepsy, but all these patients have similar phenotype, and all of them have generalized tonic-clonic seizure, focal, absence, and myoclonic. This type of mutations is variable, but high concordance because phenotype is similar. And for example, this variant, we have 2 patients with these mutations, and both have epilepsy. One patient has absence and tonic-atonic, and second patient has generalized tonic-clonic seizures. And for example, patients with this mutation has only infantile spasm. But we can see that epilepsy is more severe by patient in the patient with Argonaute mutation. And this table that show our detailed information about our patients. For example, this mutation, these family mutations, mother is carrier, and mother has monozygotic twins, and only one twin from monozygotic twins has absence, but single episodes. And these 3 patients with same mutations, 1 separate patient, and 2 sister and brother who inherited mutation from mild symptomatic mother, and both have similar phenotype. And for example, these same mutations in natural domains, both patients have absence and similar phenotype also. Treatment, patient with Argonaute mutations use similar treatment. 2 patients have special treatments like ketogenic diet, but in 3 cases, seizures ongoing. And more than half of patients have seizures free, and about 20%, 3 patients have persistent and without improvement. And 1 patient, based data from questionnaire, doesn't have any medication. And general information about epilepsy in Argonaute patients. We have about 50%. Age of onset early than Argonaute 1. Seizures type is also high variable. We have 8 cases with status epilepticus. Among patients with Argonaute 1 mutations, we had only 1 case with status epilepticus, and these it were reported in 8 cases, and in 3 cases recurrent status epilepticus. And phenotypic concordance, I was told that same mutations, patients with same mutations have similar epilepsy phenotype, and 2 case with family mutations. This 1 monozygotic twins with absence, and these 1 family brother siblings, they have similar phenotype, and 1 separate patients, but this patient has also similar phenotype. And our conclusion that Argonaute 1 and Argonaute 2 patients have overlapping clinical features, but patients with Argonaute have more severity clinical symptoms. And main features is muscle gap, hypertonia, development delay, speech delay, and others. And Argo patients have more frequency complication from another system and organs. For example, respiratory difficulty and gastrointestinal disorders. Epilepsy is same in both group, 50%, but patients with Argonaute mutation show a more severe phenotype, epileptic phenotype, and treatment are mixed, and some patients have seizure freedom, and some patients have therapy resistance. And thank you for your attention.
Hi, thank you so much. That was great. I have a question. How should we handle changing symptoms and the natural history study if we've already filled it out in the past? So if we already sent you our NHS, but there's new symptoms that arise in our kids, or something has changed, medication has changed, seizures have changed, do we fill out a new form and give it to you? Or send an email? Send an email. Okay. Thank you.
It's much easier for me.
Yeah.
Email or message in Face group. By email or message in Face group.
Okay, perfect.
Yeah,
Thank you.
both.
Let them know.
Eben, are you here? Ah, you're here.
So from the data you now have, it's really, really difficult for us to get a proper registration of his epilepsy. So what I'm curious about is, we know that he has focal epilepsy. We suspect he has absence epilepsy as well. But are there other types of seizures that you would be able to predict based on his deviation? Based on his numbers and sequence and genes, whatever you call it.
Oh, maybe these questions, not to me. This study shows that many patients have a different type of epilepsy, and the age of onset is also different, from 6 months to 17 years. And yes, we are not sure that, for example, patients who don't have epilepsy now, they will not develop epilepsy in future. And I tried to underline this independent from epilepsy from type of mutation, but we have only 30 cases. And we have 60, I have analyzed 60 questionnaire, 50%, yes, and we have only 30 cases, and this is not enough for good statistic and for good prognosis and prediction, but there are some mutations The patients who have these mutations, they have always severe phenotype, for example. For example, we can some tell that, for example, with this mutation, mostly of our patients have severe phenotype. That's why we can give some prediction. And treatment also, but in my questionnaire that I have analyzed, there are a lot of different medications and effects and results, and this information, this general overview. And we need more and more information, and we should this investigate more and more, deeper.
Like, if we should recommend EEG examination, also in the patients without clinical seizures, because my experience is that patients can have EEG abnormalities, sometimes even those that are treated later, or they can have like sleep EEG discharges. So maybe we should like, it could be maybe part of clinical screening in these patients, because the earlier you diagnose and the earlier you treat, the outcome is usually better. Because
we have 2 patients with seizure activity without epilepsy now, but they have electroencephalography activity, and we can expect by these patients in future. And in questionnaire, there is one issue about investigation, MRI and electroencephalography. But not all patients have this investigation, and not all patients send me this investigation. But when the patient sent me investigation, I can overview, I can check, and for example, there are no information in this questionnaire, but they sent me this report, and I can figure out with this report, yeah.
The actual question was if we should recommend for every child with AGO1 and AGO2 mutation electroencephalography, and the answer is yes.
Yes. No, I told that yes, in questionnaire we have this punct, and it means that all patients should undergo this investigation, electroencephalography, and we can expect it.
So Amélie will have to leave in 20 minutes. Yeah, so if there are special questions for Amélie.
I actually have a question in regards to the seizures. My daughter just had a new EEG done where they discovered a new type of seizures called ESES, electrical status epilepticus in sleep. They started steroid infusions last month, well, in July. Has any other patient had steroid infusions or had steroids and seen improvement? Because when she's been sick, they've given her steroids in the past, and I noticed early on that seizures went down, and she seemed to do better. But now with the steroid infusions, we saw a little bit of freedom. We didn't see the sleepiness look. But as the steroids have gone down, now her seizures are back up again. So are steroids something that are being looked at a lot in epilepsy?
Are you aware, Olena, about steroid therapy? Did anyone report it in the natural history study? I'm not aware of it. Steroid therapy. Yeah, so I'm not aware that anybody filled this in the questionnaire. So unfortunately, we cannot comment on that. I'm sorry. But it's really interesting to hear from you that it seems to help your child. Yeah.
Sorry, she went kind of from being almost like there wasn't a person inside. She would just kind of do what she was doing, and that was it. But as soon as the steroids went into her body, like her eyes opened up again, and you could actually see like life in her, is the only way that I can explain it. And we saw the improvement. We saw her kind of getting better, physically even. She was walking better. She started to have more, she walks funny as it is when she walks with her hands up, but she's starting to bring her hands down. She's starting to feel a little more sure of herself. And it was something that we would almost see every single time that they would give her steroids. They've, in the past, just for pneumonia, she's had a lot of pneumonia going on, and we would see short bouts of improvement. But with these high steroid infusions, it was just almost like a night and day difference between how she was prior to the moment that we left the hospital.
Okay, thank you so much for sharing this.
I just want to be sure that I understand well what the doctor said. We can help, otherwise, to fill the form, we can give blood. There is some things that we can do to help, or could you?
So yeah, actually there are several possibilities. So I think the natural study will close when? In October, so you can still fill the questionnaire in English of natural study. You can also fill the Genida web form. So if you go on the website, the novel site is active, or because we changed the website, but will be. Okay. Because it's more open questions, so sometimes you can bring novel signs that were not in the questionnaire, saying, and there are different questions about what impacts the most the quality of life of you, your children, and everything. So it could be complementary to what Olena and Davor did. We are also looking for DNA sample also to find, maybe I wrote to you, because we want to see if there is a methylation signature associated to AGO1 variant, for instance. So first AGO1, and if we find, maybe we can open to the other one. We have also an ongoing project about reanalyzing the genetic data, but we are still waiting for the ethical agreement, and I will send maybe you or send Nora and Aldona all the information about that. So there is different way. I think there is a fibroblast collections, but maybe the best is to go directly to the Coriel before then sending, because it will be more easy to distribute to everybody. So yeah, different way to help. Thank you. So maybe we can just, I don't know if you want to detail the function, or maybe just say one word about the research ongoing and maybe a summary of the conference and the team working on AGO1 and AGO2 variants. You want to do it, or you want me to do it before leaving?
If you want.
Okay. I can just say one word. I'm not sure, there were a slide just before this one. Not sure it's still, no, with the map of the world.
Yeah.
We can go back.
You can go back.
No, not this one. Okay. So it's not updated. It's the one we did at the last conference last year. Was just to show you that there are different people. So from the first conference in Regensburg in 2022, the last one in Copenhagen, and now this one in Prague, we really have more and more teams that work, that usually work or not on AGO function, and that decide to work on the variants found in the different patients. So that's teams that are really growing. So we are now a team in Europe that I work with mouse model, with induced pluripotent stem cell model. So it's cells that could be differentiated from fibroblast of patients and that could reprogram into neural stem cell. Neural stem cell or brain organoid to try to understand the effect of the variant during brain development. We have also, so maybe we have different team in, so in this slide you don't see there are different teams that are generating animal models. So you can see a C. elegans, which is a little worm, and I will show you a slide just after. And also there are novel teams that are doing a fly model. So I don't know if you know, when you have like some fruits that are becoming a bit old, you have like these little flies that can, and it's an animal model in human biology, in molecular animal biology, we say. And so they are trying to put the variant identified in patient in this fly. And so, yeah, we have different teams that try to understand with their own models, the models they are working on, what could be the consequences of the variants identified in the different patients. Where is? I cannot move. Maybe this. Okay. For instance, there is a team. Maybe he moved. Kotaro is still there, or he already left. Ah, you're still there. So maybe you won't say one word about your work in like a few seconds. It would be better than me.
Everyone, my name is Kotaro Nakanishi. So I was very shocked when I attended this meeting in Germany 2022. I have been studying Argonaute protein, but I didn't know that this is disease. But so I met this couple of the family, shocked me, and then I started thinking about what I can do. So so I'm just doing the basic science, but if even the small things maybe can show a hope or something improved, I'm very happy. So thank you.
Thank you. So he did here the structure of the protein with one of the variants, the most recurrent variants in AGO1. And we have also a team working with worms. So it was 8 years ago, I think, something like that. I think Victor left and Anna also already left. But so I met Victor Ambros in a meeting, and I presented the mutation found in AGO1, and he was working, he's still working with this small animal model, which is a small worm. And he told me, it would be really interesting to put the variant in the worm. And so that's what they did with another team in US. So both teams are in US, Victor Ambros and Anna Ielesicheva. And they introduced different variants found in AGO1 in a worm system to study what could be the consequences in worm. And maybe you are not aware, but it just won the Nobel Prize last year. And so really, it could be a good relay to really amplify, to talk about the Argonaute syndrome. And it took already a lot when he was interviewed for his Nobel Prize. So I will just keep the result, but just to show that in this little worm, when you introduce the variant found in AGO1, you will have a phenotype. You can see that the worm does not really develop totally well, and so you can study the effect of the variant like this. The other team working, so our team is also working with Argonaute syndrome. So we don't use animal model, but we are using cellular model. So we are using induced pluripotent stem cell from patient or a regular one where we put inside, we introduce the mutation in AGO to test. And we are developing them differentiating them into neurons and also cerebral organoids. So we are doing that for AGO1, and the team of Davor Lessel in Regensburg is doing that for AGO2 also. So doing that, we are trying to understand what could be the consequences of this variant during brain development, and how it could affect brain development and brain functioning also. So it's an in vitro model, but all these different models, so the worm, the fly, the molecular the structural biology, and the cellular model, all together can really help us to better understand what are the consequences of the different variants found in AGO1, AGO2, and even AGO3. I think I just missed one team, which is a team of Hans Jürgen, who developed also a mouse model for variants in AGO2, and so you can see that there are really these different organisms plus cellular model, and we are trying to join all the effort together to try to better understand this syndrome. Don't know if you wanted to add something. d'abord? No? Okay. So I don't know if you have questions about the research ongoing or other questions for us.
Maybe there was one question earlier in online where someone was asking when can we expect to maybe benefit from some treatment or new treatments? I guess that's the question. And well, that's maybe related to natural history study, but are there specific therapies that are recommended or other support?
So, I mean, currently the only therapy that I can recommend is the one for muscular hypotonia of specialized physiotherapy. This is the only thing that we really know that helps these children. The path to a personalized treatment is something that I cannot say anything about. You know, it depends on many different aspects, and it would be absolutely unprofessional if I would say any time frame.
What we can just add, it's really beneficial to have all this team working with different animal model and different cellular model, because at one point, if you have some potential clue or any potential therapeutic option, I say really potential, we will have all the model already developed, already characterized to be able to test this potential therapeutic option. So that's really our job that we are doing, and it's really useful to develop all these models.
Would it be premature to ask about stem cell therapy for treatment or helping? I found a clinic in Panama that does stem cell therapy, and I've considered for all of her issues if it would be helpful for her.
To have
stem cell therapy.
So stem cell therapy, for instance, are developed for neurodegenerative disease. So it's already a bit ongoing, like clinical trials. For neurodevelopmental disorder, is something that's
it's rather experimental, but yeah.
It wouldn't hurt.
But
we don't know.
To my knowledge, there is no neurodevelopmental disorder, even the well-known for 30 years, like Fragile syndrome, for which there is this kind of stem cell therapy that is developed. So, but we cannot say never, but it's not something that in the field of neurodevelopmental disorder, which is currently under development.
I don't want to take away time from the families, but I was wondering, is there anyone that is trying to develop a model in zebrafish?
Yes, actually. I was present last year.
Also this year, Daniel
also
Cifuentes.
This year?
Yes.
Maybe I missed this presentation, but so no. Yes, there is a zebrafish model that will be developed.
Daniel Cifuentes was here. Yes. He did not present.
No, he didn't present this year. He presented last year. Because maybe for those who are not aware, in biology, we have different animal models. As I said, the little worm, C. elegans, the fruit fly, so Drosophila melanogaster, and also zebrafish, which is a small fish that is transparent, so you can follow a lot of things inside. And so and mouse model. And basically, it's the four really organisms that are really well studied in science. So it will be good to have all this model and maybe additional model. You can imagine there are xenopus or different things that there are even, maybe I didn't mention, but there are even people working with plants, Argonaute proteins that are trying to put the variant in plant also to see the effect on the Argonaute functioning. So it's not only animal, it's also plant.
And perhaps Peter can also do some slugs?
More questions in the, I think online there's no other question. Any more questions in the room or no? Then. Thank you very much for presenting. We can switch back to the other laptop. Okay, we're done. Yeah. All right, so just about to close. Kari, did you want to close? Or, okay. Okay, so yeah, thanks everyone who was here today either to present and to listen, for those who were in the room. Also special thanks to our parents who had the courage to be on the panel and share very personal stories and how you live with the syndrome. And thanks for those online who dialed in. And yeah, we'll be sharing via email recordings, some of the links we discussed today, like the different studies. And for those who are here, we will be then moving to dinner, which is also open to the scientists, which is not far from here. And yes, also that we'll be sharing a little questionnaire just to gather your feedback, both to understand what you liked, what we could do better, but also for us then to understand or demonstrate the purpose and the impact of this conference when we will go and raise funds for the 2027 gathering. So your feedback will be much appreciated. Thank you.